Hi, my name is Brooke Donzelli. I'm a senior biology major at the University of Massachusetts Amherst at the Adler Lab, and I did my senior thesis studying the effects of Monarda fistulosa monoterpenes on the Crithidia bombi infection in Bombus impatiens. Wild and managed bees are extremely important pollinators that contribute to vital ecosystem services that help maintain human life and biodiversity. However, their populations are currently in decline, with pathogens being a contributing factor. One of the bee species infected by pathogens is Bombus impatiens, more commonly known as the common eastern bumblebee. Bombus impatiens are the most prevalent Bombus species in North America, and their populations are currently rising, so they might not need our help as much as some other bee species do. However, they are very important and convenient models when researching bee parasite interactions. One of the pathogens that infects Bombus impatiens, as well as other wild and managed bees, is Crithidia bombi. Crithidia bombi is a trypanosomatid gut pathogen, and it is known to reduce queen fitness, worker survival, and colony performance. It is also linked to the decline of many bumblebee species. Monarda fistulosa, more commonly known as wild bergamot or bee balm, is a common wildflower and attracts many pollinators, and is highly recommended for pollinator plantings. Within the species, individual Monarda fistulosa plants can have different chemical phenotypes or chemotypes. This allows individuals to produce one main monoterpene in the aerial parts of the plant, including the nectar. These commonly include thymol and carvacrol, but more geographically localized are linalool, geraniol, and eucalyptol chemotypes, and these are distinguishable by smell. One of these monoterpenes, Thymol, was found to reduce the Crithidia bombi intensity in Bombus impatiens when consumed by Bombus impatiens. However, along with Thymol, Monarda fistulosa can produce these other monoterpenes, but their effects are currently unknown. We hypothesized that these chemicals would reduce the Crithidia bombi pathogen intensity in Bombus impatiens because all of the compounds are structurally similar to Thymol. To test our hypothesis, we conducted diet assays with Crithidia bombi infected bees from 3 different commercial colonies in our laboratory. We conducted 6 rounds of the experiment, and each round was 1 week. We used 36 bees per round, and each bee was assigned to an individual nectar treatment. These nectar treatments included the monoterpenes produced by Monarda fistulosa dissolved in sucrose, as well as a monoterpene-free control solution. On the first day of our experimental week, bees were infected by Crithidia bombi, and then the infected bees were randomly assigned to one of the 6 nectar treatments to be fed for the week. After 1 week of consumption, bees were dissected and their guts were removed. We then used a microscope to count the pathogen loads in each bee. During the experiment, we also kept track of the amount of sucrose solution the bees were actually consuming. As you can see, there was no significant differences in the amount of treatment that the bees consumed. The treatments also did not have a significant effect on mortality of bees, so no treatments were causing the bees to die when being consumed. We found that Thymol, carvacrol, and eucalyptol sucrose treatments had a significant reduction in Crithidia bombi infection intensity when compared to our control solution. With Crithidia bombi count being on our y-axis and treatment being on our x, you can see that the control treatment had bees with a much higher Crithidia count than the monoterpene treatments. Geraniol and linalool had intermediate effects, but were not significantly different from either the control or from Thymol. And Thymol, carvacrol, and eucalyptol showed significant pathogen-reducing effects. Our results suggest that Monarda fistulosa plants with the Thymol, carvacrol, and eucalyptol chemotypes, as well as other plants with these nectar metabolites, could reduce infection when consumed by Bombus impatiens. It can also give rise to other studies and potential questions. We can use the system to study the effects on bee health. We could do field experiments with plants that produce Thymol, carvacrol, and eucalyptol in their nectar. This would allow us to see how plantings of Monarda fistulosa would affect pathogen prevalence across bee communities. It also gives us a convenient study system to study the possible self-medication of infected bees. For example, if uninfected bees and infected bees are given the choice between these monoterpenes, if uninfected bees show no preference towards any specific monoterpene, and infected bees show preference towards consuming monoterpenes that could reduce infection, this could be an example of self-medication. We could also ask the question if these monoterpenes could reduce infection directly in other bee species other than Bombus impatiens. We can also use this system to study the effects on plant fitness. It has been found that secondary metabolites can mediate pollination and pollinator-parasite interactions by acting as an attractant that can increase plant reproductive success while simultaneously treating infection. We can use this system to study the fitness of Monarda fistulosa chemotype in response to bee preference for a specific nectar metabolite. In conclusion, our discovery suggests that Thymol, carvacrol, and eucalyptol nectar metabolites can be used to reduce pathogen prevalence in Bombus impatiens. It also highlights the overall importance of studying medicinal floral rewards to improve bee health. I would like to thank Dr. Gordon Fitch and Dr. Lynn Adler for being such amazing mentors and for being so unbelievably helpful and encouraging during my senior thesis process. I would also like to thank the Adler Lab for being such an amazing group and such supportive people, and to the Mass URC for giving me the opportunity to share my research. Here are my references, and thank you.